And then finally, Annabelle Lyons from Edinburgh to tell us about the continuing story of Notch in cholangiocarcinoma. Thank you.
Hello, yes, I'm Annabelle. Thank you very much to the organisers of AMF for inviting me to speak today. So I think this conference has been brilliant in highlighting all the different complexities within cholangiocarcinoma research at all different stages, at the patient kind of diagnostic level, at the treatment level, and also at the scientific level and the cellular level. So today I'm going to be talking about a particular cell signalling process, the Notch signalling pathway, and how through this work we've shown that it drives the initiation and early growth of cholangiocarcinoma. So what is Notch signaling, for those who haven't come across it before? It's an evolutionarily conserved pathway of cell-to-cell communication. It happens at short range between neighbouring or opposing cells, and it ultimately leads to transcriptional changes, which are gene expression changes, in one of the cells. This can ultimately lead to a variety of cell fates, including cellular differentiation, proliferation, and cell death, apoptosis. We also know that this particular pathway is important in tubular morphology and patterning. So when we think about the bile ducts as biological tubes, which is what they are, this pathway is quite important. So in the human Notch system, it's quite actually mechanistically very simple. It has 9 core components. These are 4 receptors and 5 ligands. So these receptors and ligands act like a key in a lock. They can combine in various conformations at the cell surface of cells and lead to different outcomes. So the first step of the Notch signaling pathway is signaling. So a ligand expressed on a signal sending cell will bind to a receptor on a signal receiving cell. The binding of this receptor to the ligand actually causes a conformational shift in the receptor to expose an enzymatic cleavage site called site 2. Site 2 is then cleaved by ADAM proteases, which are extracellular proteases that cut the Notch receptor and lead to the release of the extracellular domain of the Notch receptor along with its bound ligand, and leaves behind this truncated form of the Notch receptor. This truncated form is then cut again, this time by a different group of enzymatic proteins that form up this complex called the gamma secretase complex in a process called S3 cleavage. So this S3 cleavage is actually really important. It releases the only direct signaling molecule on the Notch signaling pathway, which is the NICD, or Notch intracellular domain. The NICD translocates to the nucleus, where it binds to a transcriptional regulatory complex of proteins. One particular protein that I wanted to highlight is called CSL in humans and RPPJ kappa in mice and other model organisms. So this protein is really important because in the absence of the NICD, so when Notch signaling isn't happening, it actually recruits transcriptional repressor proteins. These supercoil the DNA, shut it down, and inhibit access by transcriptional machinery. However, when the NICD is present, it dispels these transcriptional repressor proteins, recruits transcriptional activators, and leads to the activation or the gene expression of Notch target genes. So why look at this pathway when we think about bile ducts? Well, work from many laboratories over the past few years have shown that it's a master regulator of biliary form and function. If we look at the developmental context for a minute, during embryonic liver development, progenitor cells called hepatoblasts can become one of 2 cell fates, and one of these is a biliary epithelial cell, so a cholangiocyte. To become a cholangiocyte, it has to express the Notch2 receptor, one of these 4 human Notch receptors. This Notch2 is provided with cognate ligand JAG1 by the surrounding portal vein mesenchymal and endothelial cells. And the importance of this Notch2 JAG1 signaling access in biliary specification and in the establishment of the bile ducts early on is showcased really well by a developmental disease called Alagille syndrome. So in Alagille patients, particularly in their livers, they completely fail to form bile ducts. And Alagille syndrome has been attributed only to mutations in either JAG1 or Notch2. So perturbing either of these factors leads to total lack of formation of the biliary network. We know that they're important early on. So if we leave development behind now and only focus on the adult context and the adult disease, we know that upon biliary injury and hepatic injury, which we sustain throughout our lives, we get reactivation of this developmental process, Notch signaling, particularly in cholangiocytes, so biliary epithelial cells. This aids in the repair and regeneration of the biliary network. And this is a homeostatic or normal process. We also know in intrahepatic cholangiocarcinoma, I'll be calling it ICCA for the rest of this talk, in ICCA, particularly in tumours extracted from patients at late stages of the disease through biopsies or from resected livers, that we get this persistent Notch activation signature. So these tumours have overactivation of the Notch signalling pathway. What we don't know or didn't know at the start of this project, was what happens in the interim, what happens at the transition point from pre-malignant biliary disease into cancer, and is the Notch signaling pathway involved? So my research questions were kind of multifold, focusing on this transition state between pre-malignancy and ICCA formation. Firstly, can this pathway, Notch signaling, drive ICCA formation, particularly looking at the biliary epithelial cells themselves? So there's a mouse model of ICCA formation called the NICD-AKT mouse model. And in this model, we can drive the transdifferentiation, so the change in cell state from hepatocytes into cholangiocytes, so biliary epithelial cells, through activation of the Notch signaling pathway. This is well established and well known. What we don't really know is whether Notch signaling drives ICCA formation from the biliary cells themselves in a cell autonomous fashion. If this is the case, that Notch signaling drives this process from these biliary epithelial cells themselves, which receptors and ligands are responsible? Who are the key players in this process at the upstream end of the pathway? And also looking downstream, what are some of the downstream genetic targets that are activated and cellular pathways that are activated that can then go on to allow cancer to form? And could we intervene at either of these stages, either upstream in the pathway or downstream, with therapeutic benefit for patients? Lastly, as a lab, we're really interested in how bile ducts grow in all contexts, both health and in disease. So I wanted to ask whether Notch signaling orchestrated the growth of these tumours and how it did that, particularly in the context of pre-malignant biliary disease. And I used human patient samples to answer this last question. So in the lab, we rely heavily on mouse models of ICCA formation. So why look at mice in general? Models are really powerful because they allow us to model what's happening at the earliest stages of the formation of this cancer. We know from the stories that we heard in the last few days that cholangiocarcinoma is often caught really late. That means that our access to patient tissues is also at late stages of the disease, looking at advanced cancer. We don't have almost any patient specimens that show us what's happening early on in the formation of ICCA. So mouse models allow us to do that. In the lab, Luke, my boss, generated a mouse model called KPP-TOM, which stands for keratin 19, TRP53, P10, and TD tomato. In KPP-TOM mice, it's quite an elegant model. They form their bile ducts normally. They go through adolescence and reach adulthood, and only then can we induce the loss of expression of two oncogenic repressor genes. These are P10 and P53, both of which have been found mutated or acquired as mutations in adult patients. So when we lose these factors, we can lineage trace which biliary epithelial cells have lost P10, P53 expression through TD tomato protein activation. So these cells go red. So what was interesting is we noticed that if we leave this mouse model alone, no cancer forms. So just the genetic mutations weren't enough to cause cancer. However, if we additionally added in hepatic injury in the form of treatment of these mice with a hepatotoxin called thiacetamide at a low dose in their drinking water, cumulatively over time, the result was very different. So what we noticed, here's some quantification looking at tumor burden in these animals, 3 mice in each group, that if we didn't treat these animals with a hepatic damaging agent, that's the week 0 seen in this graph, no cancer forms at any time point. However, if we cumulatively treat them 2 weeks, 4 weeks, 6 weeks, up to 8 weeks with thiacetamide, we get a continuous accrual of tumor burden. And we also had a control group where we didn't give these animals any hepatic injury and left them for 6 weeks and no cancer formed. So in this model, we require both tumor suppressor loss and hepatic injury in the inflammatory microenvironment this produces to form ICCA. I analysed this model looking at what happens at various time points. So in the left-hand side here, you can see untreated mice. So these are mice that have lost P10 and P53, have transformed cells, but haven't had any hepatic injury. In these animals, you can see the transformed cells in brown here, but the morphology of these ducts are normal. By 2 weeks of thiacetamide treatment, you got proliferation of these ducts, but no obvious cancer. And it's at this 3-week time point that we start getting the earliest incidence of morphologically distinct cholangiocarcinoma forming. So I was interested in looking at what the Notch signaling pathway was doing here, particularly what changes between 2 weeks of thiacetamide treatment and 3 weeks, mimicking this transition point between pre-malignant biliary disease and cancer formation. So the first question I talked about earlier was what receptors and ligands are involved in the Notch signaling pathway, and are these involved in ICCA formation? I set up an experiment, an RNA sequencing experiment, with 4 animals from this mouse model and treated them with the hepatic damaging agent TAA for 3 weeks. I isolated the TD tomato positive cells. These are the ones that have loss of P10, P53 expression. and first looked just at the raw recounts of the different Notch receptors and ligands. Interestingly, there were only 4 factors that were expressed at all. 2 receptors and 2 ligands. The receptors were Notch1 and Notch2, and the ligands are Jagged1 and DLL1. The factor that was most expressed by a country mile was Notch2, and I'll come back to this in a minute, why we think this is important. But we wanted to interrogate, now we know who our key players might be, we want to look more specifically about when they're expressed, how they're expressed, and what they're doing. I took advantage of a single-cell RNA sequencing dataset generated by another postdoc in the lab, Ed Jarman. He set up an experiment with 3 groups of animals, and for simplicity's sake, I'll just say what the kind of overall differences in these groups were. One group had only hepatic injury. One group, the second group, had only genetic mutations, and the last had a combination of both, which we know leads to this tumor-permissive state where cancer forms. So he extracted the biliary epithelial cells from mice from each of these 3 models and compared their transcriptomic signatures. So what are the differences in gene expression in these 3 groups? And generated this UMAP. So all the cells you see here are cholangiocytes, but different types. So you see large duct cholangiocytes in 2 clusters, small duct cholangiocytes in 1 cluster, and then a proliferative cell population as well. And at the bottom, you can see the cells that cluster from hepatic injury alone, tumor suppressor loss, or a combination of the 2, leading to this tumor-permissive environment. And you can see that this is kind of an amalgam of the previous 2 graphs. And I want to focus, because of time, I want to focus only on one particular subset of biliary epithelial cells that had a very different change in expression, which are the large duct cholangiocytes. So focusing on this particular part of the UMAP, what was interesting is that we see between these 3 groups, they segregated based on genetic status. So the pink cells here are wild-type cells. They've undergone hepatic injury, but these are cells that don't have any genetic changes. The cells on the right-hand side are groups are the genetic mutations only group and the genetic mutations plus damage group. So the only difference between these 2 are whether they have lost P10 and P53 expression and have this susceptibility to forming cancer, or are wild-type genetically. And when we look at the 2 ligands that I spoke about earlier, expressed in our ICCA mouse model, both of these only clustered in the group that have P10 and P53 loss. This is telling us that Notch ligands are enriched in biliary epithelial cells that have lost tumor expression, tumor suppressor gene expression. So then looking a little bit about the receptors, I did some immunofluorescence on animals either that had been untreated with TAA or treated with TAA, and tried to look at the expression of the various Notch receptors, and focusing on Notch2 here. What's interesting is in these immunofluorescent images on the left-hand side, TD tomato shows the cells that have lost P10 and P53. From the data I just showed you, we know that that means that they have an increase in Notch ligands. However, they have Notch2 expressed by all biliary cells here, and this is in the context of normal bile ducts that are uninjured. When we add in TAA into the equation, we start damaging these biliary cells, we get acquisition of these neoplasms, these early cancerous lesions. And we think this has to do with the first tumor suppressor loss, then activation of the ligands, and now injury, allowing this kind of proliferative state. If we allow these mice to be treated additionally with TA for longer, we then get establishment of these ICCA tumours. And this is because we've acquired this ligand-rich niche. But what's important to mention is that all biliary cells in the context of health and in early ICCA and then in established ICA all express Notch2. So basically, if you're a biliary epithelial cell, you'll express this receptor. What we can't tell from these images is whether this receptor is just expressed or whether it's being activated. So I'll come back to this in a moment. But if we zoom in on this last panel, so looking at ICCA tumours, we can see something quite interesting, which is that Notch2 is expressed basolaterally or laterally between cells at the cell junctions between these cholangiocytes, not at the basal membrane as we see in the developmental context. So it seems like all biliary epithelial cells are actually expressing these receptors only facing each other in a cell autonomous fashion. We crossed the Cape-Piton mouse line with another one where we can inducively overactivate the Notch signaling pathway. So in these mice, the red cells are still demarcating loss of P10 and P53, but now we have these positive GFP positive nuclei, which also tell us that in these cells we have overactive Notch signaling. I set up an experiment with this mouse model where I induced the loss of P10 P53, overactivated Notch, and then I left these animals long term. I didn't give them any hepatic injury. And what was interesting is that in this group, I was able to quantify that tumours formed 100% of the time. However, in normal KPP-Tom mice, where we need this hepatic injury and inflammatory environment, we didn't see any cancer forming. So there's something about Notch which allow, on the background of tumour suppressor loss, which drives ICCA initiation without the need for hepatic injury. Also, the morphology of these tumours was quite interesting. They all seemed to be large duck-like in morphology, and they were hyper-folded as well. So I set up another experiment trying to look now at growth of these of ICCA in these models. So I treated both KPP-Tom and KPP-Tom NICD mouse lines with TAA for 3 weeks and compared and contrasted both tumour count, which is the raw number of tumours, as well as tumour burden, which is the percentage of the liver lobe covered in tumour, so the invasion of the liver, and found that there was a significant increase by both metrics in the KPP-Tom NICD model, so the one that had overactive Notch. I also wanted to characterise where exactly proliferation was happening in these tumours. The white cells you can see here are ones that are proliferative, and we saw a significant increase in proliferating biliary epithelial cells as a result of overactive Notch signalling. The next thing I noticed when looking down the microscope was that a lot of the proliferative cells seemed to be clustering at the outer or leading edge of the tumour. So I wanted to quantify this scientifically. I used a quantification tool called QPath, and you can see some of the analysis images here where the red cells are the ones that are proliferative. And quantifying these, I could see that basically in all cases of tumour growth, we get a leading edge or proliferative ring at the outside edge of the tumour. So it's growing from outside out, not from kind of proliferative core. In addition, if you quantify the number of proliferative cells as like a percentage of the total number of cells per each tumour, regardless of the tumour size, in KPP-Tom mice that had damage, KPP-Tom NICD mice without damage, or KPP-Tom NICD mice with damage, so Notch signalling and hepatic injury, we can see that in this latter group, we have a significant increase in proliferative cells to about a quarter of all cells within the tumour. So we now know from the data that I've showed you that we get an increase in the ligands when we lose tumour suppressor genes. We also know that we get overactivation of the proliferative pathway and kind of overgrowth of these tumours. But we wanted to ask a little bit more about who are the, looking at the mechanism of this. So how does Notch2 and its ligands kind of contribute to growth of the biliary epithelium? So we then turned to looking at human premalignant disease. One particular disorder, a disease called IPMB, intraductal papillary neoplasm of the bile ducts, is a variant of bile duct carcinoma, so it's a biliary tumour, but it's premalignant. It doesn't breach the basement membrane. We sourced this material from the Scottish Tissue Biobank Archive, and when I stained using a antibody that recognises the cleaved or active version of the Notch2 receptor, which I couldn't use for mouse tissue, frustratingly, but did work in human tissue, we could see that it's only the IPMB epithelium, so diseased epithelium up here, that expresses the cleaved version of Notch2. So this is showing that Notch2 is activated in these cells only. So even though all biliary epithelial cells are primed with Notch2 receptor, it's only cleaved in this disease state. Also looking at the ligands, we can see that in large duct cholangiocytes we get JAGAD1 expression, but in these kind of smaller ducts, both in health and some in the disease, we get DLL1 expression. So different ligands are being expressed in different contexts here. I also made use of something called a TMA, which is a tissue microarray. So these are, Judith mentioned yesterday about having biopsies. These are those biopsy tissues and how we can use them scientifically. So I had some control tissues, some IPMB tissues biopsies, and some ICCA biopsies, and wanted to quantify the number of Notch2 positive proliferative cells to show, and found that there was a significant increase in proliferative cells both in IPMB and ICCA. So the last bit of my talk today is going to highlight about the downstream targets of Notch signalling and whether any of these could serve as particular factors that we could mitigate when thinking about potential therapeutic interventions for patients. So what are some of the downstream targets in ICCA formation? We've set up another RNA sequencing experiment comparing animals from the KPP-Tom and KPP-Tom NICD lines after 3 weeks of TA treatment, and found a whole host of both up and down regulated genes, many of which are known to be Notch target factors. There are three particular ones I wanted to highlight here, and I'll talk about in a moment, which have all been described in various cancer situations, but not in iCCA, and they're not very well characterized in general. So the first is CSPG4. It's a proteoglycan. It's been described very well in melanoma, particularly in the invasion and metastasis of this cancer, and it's associated with inflammation. It's not actually expressed in normal conditions. We will have another one called NRAP, which is a downstream effector of Notch. It's involved particularly in regulation of liver cell self-renewal. And lastly, HAL, which is a classical downstream effector of Notch, but it's also an activator. So it helps, it's upregulated by the Notch signaling pathway, but also is involved in shutting it down. So it's helping the spatiotemporal control of Notch signaling and informing cell fate decisions. So we set up an experiment to try to interrogate these Notch downstream genetic targets in cholangiocarcinoma using this model I mentioned very briefly earlier called the AKT and ICD mouse model. So in these mice, we can activate iCCA formation from hepatocytes, driving their transdifferentiation into biliary epithelial cells. And because this model is Notch dependent, it was perfect for us to ask the question, if we knock out these various potential Notch target genes in this process, do we get a change in the number of tumours that are forming and also in the tumour burdens, the amount of cancer that we get? So along with a master student, we set up this experiment knocking out these three genes and also had two control groups. One is a scramble control, so we're not knocking out anything, and then a positive control group, which is for RBBJ kappa, this gatekeeper gene, and also HES1, which is a very well characterized downstream Notch target. These, we knew that perturbing either of these factors would change tumour number and burden. So this is just proving that it works. We get cells that are keratan-19 positive, means they're biliary. They have CFP expression, which means that they've taken up our constructs to knock down these various genes. And if we quantify the tumour count and the tumour burden, we get a significant increase in one particular group, which is CSPG4. So I'm now doing work to characterize how this factor mechanistically informs iCCA growth. So the summary of my work here and our current hypothesis is that, at least in part, we have a Notch-dependent malignant transformation of the biliary epithelium in iCCA. In the normal biliary epithelium, throughout life, we can acquire pro-oncogenic mutations. However, unless we have hepatic injury, they don't really seem to instigate or activate. So we get this activation of Notch ligands. With continuous hepatic injury over time, we get proliferation of these cells, forming kind of dysplasia and an acquisition of this Notch ligand-rich niche. Intracellularly, we get expression of particular target genes, and the next steps are trying to define whether if we perturb any of these, we could actually have therapeutic benefit. So big thank you to all the members of the Bolter Lab, the members of the IGC, and the various groups that have helped us get this work done. Thank you.